Dear students, today's topic is study on diseases of brinjal and vindi. First is diseases of brinjal. Brinjal is one of the most commonly grown vegetable crops of the country. India produces about 7.676 million metric ton of brinjal from an area of 0.472 million hectare with an average productivity of 16.3 metric ton per hectare. Its production is greatly reduced by many diseases like Cercospora leaf spot, Alternaria leaf spot, bacterial wilt, little leaf of brinjal. So, let us discuss about the fungal diseases. First one is the Cercospora leaf spot. This disease can be characterized by isochlorotic lesions, angular to irregular in shape, later turning greyish brown with profuse sporulation at the center of the spot. Now coming to the scientific classification of this pathogen. Kingdom is fungi, phylum Ascomycota, class Dothidiomycetes, subclass Dothidiomycetidae, order Capnodiales, family Mycosphaerillaceae, genus Cercospora, and species is Melongena. Now coming to the character of the pathogen. Conidiophores born singly or in fascicles, arising from stroma or directly from the vegetative hyphae. Pale brown septate or non-septate, which are hyaline to dark brown pigmented, continuous, simple or branched, uniform to clavate, sometimes obclavate, curved, smooth, conidia borne terminally and singly, becoming lateral by sympodial development of the conidiophore, acicular to obclavate or cylindrical, rarely clavate, thin-walled, multiseptate. The conidiophore has a subapical growth, reason for the mature conidium to push aside, which led to geniculations bearing scars at the area where conidia are detached. Conidiophore with distinct geniculation, that is knee-like bands, which is an important character for identification. Due to geniculation, conidia are formed singly, acrogenously, that means at the tip. Conidia 4 to 10 celled, multiseptate, hyaline or brown conidia. Conidia measuring 63 to 140 into 14 to 17.5 micron. This is about the character of this pathogen. Next disease is the Alternaria leaf spot. This disease can be visualized only when spots with distinct concentric rings bands of light and dark colors, irregular 4 to 8 millimeter in diameter, and may coalesce to cover large areas of the leaf blade. Fruits causing large deep-seated spots and drop off prematurely. Now coming to the scientific classification of this pathogen. Kingdom is fungi, class Dothidiomycetes, subclass Pleosporomycetidae, order Pleosporales, family Pleosporaceae, genus Alternaria, and species is Solani. Next is the character of the pathogen. Mycelium is septate, branched, light brown to dark brown. It has inter and intracellular. Conidiophores emerge through stomata and dark colored. Conidia are single-celled, muriform, conidia with non-catenate, which means solitary conidia with long filiform beak. The conidial septation with 5 to 10 transverse septa and a few longitudinal or oblique septa. Next important disease is the root rot. The disease can be characterized when underground portion, that is the root portion, causing rot. Next is the scientific classification of this pathogen. Kingdom is fungi, phylum Ascomycota, class Sordaromycetes, subclass Hypocreumycetidae, order Hypocreales, family Nectriaceae, genus Fusarium, species Incarnatum. Now coming to the character of this pathogen. Fusarium has a hyaline septate hyphae, short conidiophores. Septation with a terminal phyllite may be simple or branched. Conidia are formed at effuse sporodokia called pinnodes. Conidia are of two types: microconidia, which is single-celled, oval or comma shape, pyriform or elongate, and macroconidia, which has hyaline, spindle shaped with pointed ends. It also may have 3 or more septation, distinct pedicellate base. Macroconidia has a distinct feature of having foot cell. Macroconidia straight, seldom curved, 3 septate, 24 into 3.5 micron, 7 septate, 35 to 52.5 micron into 3.5 to 5.2 micron. Microconidia oval shaped, 1 or 2 celled, thin walled. 0 septate size is 4 to 6 micron into 2 to 6 micron. 1 septate size is 8 to 20 micron into 2 to 4.5 micron. Chlamydospores are formed from the hyphae or at macroconidia. So this is about the character of this pathogen. So next is the bacterial disease. First one is the bacterial wilt. Bacterial disease mostly show wilting, stunting, yellowing of the foliage, and finally collapse of the entire plant are the characteristic symptoms of the disease. The vascular system becomes brown. Bacterial ooze come out from the affected parts. Now coming to the systematic position of these bacteria. Domain is bacteria, phylum Proteobacteria, class Gamma Proteobacteria, order Pseudomonadales, family Pseudomonadaceae, genus Pseudomonas. Now coming to the character of this bacteria. The bacterium is non-acid fast, non-spore forming, non-capsulated and motile by a polar flagellum. The bacterium produces acid but no gas in dextrose, sucrose, lactose and glycerol. Starch hydrolyzed with slight liquefaction of gelatin. This is about the character of this pathogen. Next disease is the little leaf of brinjal. Peculiar character is infected plants show a bushy appearance. Mostly there is no flowering, but if flowers are formed, they remain green. Fruiting is rare. Now coming to the scientific classification of this pathogen. Kingdom is bacteria, phylum Tenericutes, class Mollicutes, order Mycoplasmatales, family Mycoplasmataceae, genus Mycoplasma. Now let's see the character of this pathogen. Little leaf was first considered a disease caused by a virus. In 1969, it was attributed to a Mycoplasma-like organism closely related to aster yellows and curly top. This organism having ovoid or spherical body. These measure 40 to 300 nanometer in diameter and lack a rigid cell wall. The phytoplasma are present in phloem sieve tubes. It is a self-transmissible disease. The organism has been transmitted to datura, tomato and tobacco. It is occurs in nature on datura. Next is the diseases of bhindi. Okra, popularly known as bhindi or ladyfinger, is one of the important vegetables grown throughout the country. It is noted for its rich iron content and nutritive value. For the commercial cultivation of okra has received a great setback under Indian conditions due to attack of different diseases caused by fungal, bacteria and virus like powdery mildew, wilt, leaf spot and YVMB. So let's discuss one after another. So let's discuss about the fungal diseases. First one is the Fusarium wilt. This disease appears as yellowing and stunting of the plants, followed by a wilting and rolling of the leaves, and finally the plant dies. Next is the scientific classification of this pathogen. Kingdom is fungi, division Ascomycota, class Soderomycetes, order Hypocreals, family Nectriaceae, genus Fusarium, species Oxysporum. Now coming to the character of this pathogen. The disease is caused by Fusarium oxysporum, F species Vas infectum. The mycelium is hyaline and intracellular in the host. Macroconidia are mostly 3-septate and microconidia are 0 to 1 septate. The microconidia are 5 to 12 into 2 to 3.5 micrometer, while macroconidia are 40 to 50 into 3 to 4.5 micrometer in size. Macroconidia are fusiform, falcate, curved and formed on sporodokia and pinnodes. In mass, these conidia appear buff to Solomon orange in color. Both intercalary and terminal chlamydospores are formed, which are broadly ovate. Next disease is the Cercospora leaf spot. This pathogen produces indefinite leaf spots but grows as a sooty mold on the lower surface of the leaves. Now coming to the scientific classification of this pathogen. Kingdom is fungi, phylum Ascomycota, class Dotydiomacetes, order Capnodials, family Mycospiralaceae, and genus is the Sarcospora. Now coming to the character of this pathogen. The disease is caused by 3 species of Sarcospora like Sarcospora abelmosi, Sarcospora hibicina, and Sarcospora malaiensis. These species differ in their size of conidiophores and conidia. In Sarcospora abelmosi, the conidiophores are long, brown, and bear pale olivaceous, slightly tapering conidia. The conidiophores of C. hibicina are extremely long, sometimes up to 1000 micrometer in size, narrow, and bear conidia that are sometimes hyaline and appreciably more narrow than those of C. abelmosi. The conidiophores of C. malaiensis are borne in clusters of 5 to 20 and bear conidia which are colorless, narrow, long, and tapering from the blunt base to the sharp tip. So, this is about the character of this pathogen. Next disease is the powdery mildew. Powdery mildew is an important disease of this crop and under favorable weather condition causes significant yield reductions, particularly if the infection takes place in early stages of plant growth, produces minute discolored patches with thin fine meshwork of white mycelium arising at many places on the surface of the lower leaves. The conidia, after landed on the host surface, germination through the germ tubes, elongate and form an appressorium at the tip in the initial stage of infection. Then the penetration hyphae arise to penetrates the epidermal wall, which enters the cell. Then the bulbous base finger-like projections called haustoria arise. Now coming to the scientific classification of this pathogen. Kingdom is fungi, phylum Ascomycota, class is Luteomycetes, subclass Luteomycetidae, order Erysipelas, family Erysipeaceae, genus Erysipeae, species Psychoracearum. Now coming to the character of this pathogen. The disease is caused by an obligate parasite Erysipeae Psychoracearum. The conidia are single-celled, hyaline, barrel-shaped, and in long chains. The conidial dimensions vary with the physiologic race and the host. Pleistocyathea are globose, dark with hyaline to dark brown, and mycelate appendages. They contain 8 to 18 asci, and the asci are pedicellate, ovate to broadly ovate, or ellipsoid. The number of ascospores per ascus is usually 2, rarely 3. The ascospores are one-celled and hyaline, oval to sub-cylindrical. This is about the character of this pathogen. Next important disease is the yellow vein mosaic. The characteristic symptoms include vein yellowing and thickening of leaves, forming a network of veins and veinlets in the infected leaves. Now, let us see the classification of this disease. It is grouped under group 2 single-stranded DNA, family Geminiviridae, genus Begomovirus. Now coming to the character of this pathogen. The disease is caused by yellow mosaic virus and belongs to the Bigemini virus. The size of virus is 18 into 30 nanometer. Virus particles are spherical, isometric, measuring 28 to 30 nanometer. So now coming to the conclusion of this topic. Brinjal and bhindi is to attack by several diseases such as fungal, bacteria and viruses. Its production is hampered by the several factors like biotic and abiotic factors. So, the management of the diseases can be obtained only if we understand about the character of the pathogens and as well as the symptoms involved.